Hello everyone. Welcome to today's live broadcast, Access Key Technologies to Accelerate Genetic Testing in Your Laboratory. I'm Rupert Yip of Natera, and I'll be your moderator for today's event. We are delighted to bring you this educational webinar series presented by Labroots and sponsored by Natera. Natera is a market leader in women's reproductive genetics testing. Natera's complex technologies has been proven clinically and commercially in the prenatal testing space, and the company is actively researching its applications in the liquid biopsy space for developing products with oncology applications. Since 2009, Natera has launched 7 molecular diagnostic tests, many of which are available through major health plans, accounting for more than 140 million covered lives in the United States. The company's own robust laboratory processes thousands of genetic tests per month. For more information, please visit natera.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the screen by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical questions viewing or hearing this presentation, please click on the support button at the top of your presentation window or submit your problem through the green Q&A button lower left. I would now like to introduce today's speaker, Samuel Silk. Sam received his Master's in Science in Bioengineering from the University of California, San Diego, in La Jolla, California. Subsequently, he has held scientific support positions at Thermo Fisher and Fluidigm, working with customers from a wide variety of industries. Currently, Sam is a field application scientist with Natera, providing technical assistance worldwide to labs that are implementing Natera technology to offer genetic testing, including NIPT. To better craft the presentation today, we'd like to poll the audience with a few questions. How would you describe the size of your lab? Is it 1 to 2 people, 3 to 20 people, 21 to 50 people, or more than 50 people? Select one of these and click Submit. A few more seconds. All right, the next question. All right, I see that most people are 3 to 20 and a mix of folks that are in organizations that are more than 50 people. Excellent. All right, let's move on to the next question. What type of affiliation is your lab? Are you affiliated with an academic core lab, an academic research lab, commercial testing lab, or a hospital lab? We'll provide about 10 to 15 seconds for folks to answer this question, and then we'll share the results. Okay, all right. Majority commercial testing lab, excellent, with a good representation of academic research labs. Excellent. The next question. Please rank in order of importance for an NIPT test. When you're considering an NIPT test, do you consider clinical performance, number and quality of scientific publications, or ease of use, minimal hands-on time, as the most important factor when considering an NIPT test? All right. Well, it's no surprise here. Clinical performance is a minimum, but it does look like some people are interested in something that is easy to use as well. All right. Next question. What level of bioinformatics expertise do you have in your organization? None, some but limited, moderately capable levels, or a team of expert bioinformatics staff? Okay, we've got a pretty broad mix of folks. All right, so without further ado, I'll now turn it over to Samuel Suk for his presentation, Access Key Technologies to Accelerate Genetic Testing in Your Lab.
Thank you, Rupert. Hi, everyone. Thank you very much for joining Natera's webinar today. My name is Samuel Suk, a field application scientist with Natera, and it's my privilege to be presenting to you today. So to begin, I'm going to introduce our company, what we do, and provide an overview of genetic testing with respect to NIPT. This will then lead us into our unique product offering, Constellation. So who is Natera? Natera was founded in 2004 by Dr. Matt Rabinowitz, who comes from a background in engineering, signal processing, and informatics. Dr. Rabinowitz has built a team of world-class scientists, geneticists, and bioinformaticians to deliver the world's most complex qualitative method for the detection and differentiation of fetal DNA from maternal DNA. This method is the only method available in the U.S. market that can reliably differentiate the mother's DNA from fetal DNA for genetic abnormality risk assessment. This powerful new technology has proven so popular and successful clinically that despite being the last NIPT technology to market, it has become the market leader in the U.S. For the last 6 years, Natera's scientists and engineers have been busy innovating women's reproductive genetics. At an unprecedented rate of almost one new product per year, Natera has launched highly accurate, high clinical performance tests for a number of different products, including pre-implantation genetic testing, miscarriage testing, paternity testing, carrier screening, which screens for up to 274 disease conditions, our market-leading NIPT test, Panorama, that includes screening for microdeletions, scalable bioinformatics powered by our Constellation software, and coming soon, oncology screening applications via blood draw. While we started out in women's reproductive genetics, we're applying our innovative SNP-based technology to identify heterogeneous tumor DNA that circulates in the blood of early-stage cancer patients. We believe our advanced technology is powerful enough to detect tumors with greater sensitivity and specificity over current technologies. While we are a strong player in the U.S., Natera already has a worldwide presence of over 60 countries to date, and we're still expanding at a rapid pace. So the completion of the Human Genome Project ushered in a new era in genetic testing, and with the rapid decline of the cost of sequencing, more labs are now able to offer genetic testing. Many of today's hospital and reference labs started out doing biochemical test panels and still analyze numerous samples using biochemical methods. But because of the stochastic nature of enzymes, many of these tests have low sensitivity, specificity, and positive predictive values. Further, these methods can sometimes be time-consuming, especially if they require a growth phase to generate enough sample. Amniocentesis, for example, requires a growth phase to accumulate enough genetic material for analysis. And unlike traditional biochemical tests, genetic tests can be performed much faster and with greater precision. And as genetic tests become more prevalent and have positive health economics, more private and public payers are beginning to reimburse for such tests. However, the decision to move into genetic testing isn't always so simple. It's fraught with several challenges. For example, the financial challenges of high cost of capital equipment purchases and maintenance of these equipment, complex workflow that can be daunting to next-gen sequencing novices, and highly complex bioinformatics that require high-performance computer resources as well as bioinformatics skills. And finally, to make a business case work, you need to identify a source of revenue, private, public insurance, patient out-of-pocket, for your new genetic testing service. Of all the genetic tests the genetic conditions that can be tested for, NIPT genetic testing is an ideal first genetic test to implement in a lab that's considering offering genetic testing. And NIPT is an ideal first genetic test because of several reasons. There's a very large market for such tests, and while relatively new, NIPT is already a proven technology that's head and shoulders superior to the traditional maternal serum screening method. Unlike sequencing and interpreting entire human genomes worth of data, the bioinformatics to NIPT is relatively simple. And finally, many health insurance companies and some national healthcare systems are already reimbursing for NIPT. So many labs endeavoring into genetic testing will start out with NIPT for the reasons above. And as they get more and more familiar and comfortable with this technology, they can start expanding into other genetic testing applications, such as oncology. So I'd like to spend some time to introduce you to some of the basics about NIPT and background of existing technologies. So to start, what is NIPT? NIPT examines placental DNA, also referred to as fetal DNA in many cases, in a mother's bloodstream to determine whether a developing baby is at higher or lower risk for certain chromosomal or subchromosomal genetic disorders. These may be caused by entire missing or extra chromosomes called aneuploidies, or small missing pieces of chromosomes called microdeletions. And a simple, non-invasive blood test from the mother provides an accurate screen of the baby's genetics as early as 9 weeks gestation. NIPT is actually offered by numerous companies using a variety of techniques. While most are using a method referred to as a counting method, Natera uses a different technology using SNP-based targeted sequencing. So what is different between these two technologies, and what are the benefits of using SNP-based targeted sequencing? So in counting, all genetic material in the blood is analyzed without differentiation between what is maternal DNA and what is fetal DNA. In targeted sequencing, carefully selected SNPs on the chromosomes of interest are analyzed, which allows for the differentiation between the maternal DNA and the fetal DNA. And this is a very important point to keep in mind. And in counting, where one can't distinguish the fetal signal, the observed amount of DNA is compared to the expected amount of DNA for the chromosome of interest, which is then assessed against the reference chromosome. So for example, in the case of trisomy 21, the amount of chromosome 21 is quantified against the reference chromosome 3, which is typically present at the normal 2 copies. So this results in a lower sensitivity at lower fetal fractions. Example of what such a quantification might look like is shown on the graph on the right, where in the context of maternal DNA represented in gray, the green represents the expected amount of DNA for fetal disomy, and blue represents the expected amount of DNA for fetal trisomy. Differences in the expected amounts of fetal DNA can be used for analysis of the disomy versus trisomy of the chromosomes of interest. Recall that in actuality, since there is no differentiation between fetal and maternal DNA, we now represent the fetal component in gray as well. At higher fetal fractions, the difference is larger, becoming less and less discernible at lower fetal fractions. So as you can see, it becomes increasingly difficult to discern a state of disomy versus trisomy if there is no color coding, as is the case with the counting methods. Unfortunately, we don't know whether the DNA is maternal or fetal, and must rely simply on the difference in quantity. And as such, it's vitally important to distinguish and report fetal fractions along with any aneuploidy determination. Not doing so is akin to a trial witness claiming identification of a suspect without indication of how foggy the conditions were when he saw the suspect. Before venturing into what the contrasting technology is, I'd like to briefly describe what SNPs, or single nucleotide polymorphisms, are. At Natera, we use a SNP-based technology versus a counting method as previously discussed. The SNPs are single base pair differences that occur as normal variations between different people. Although, say, 99% of everyone's DNA is the same, 1% can be different. We focus on that 1% of the DNA that can in fact be different. At some of these points, mom and baby will be the same, but at some of these points, they will be different. And we use over 13,000 of these points where they are different to determine a fetal DNA pattern within the maternal blood to determine the ploidy of the chromosomes of interest. Before getting into the specifics of how SNP-based targeted sequencing works, first a little bit of background. Humans normally have 2 copies of each chromosome for a total of 46. We target SNPs on chromosomes of interest, namely 13, 18, 21, and the sex chromosomes, to study their fetal signal. For simplicity, let's look at a single pair of chromosomes. Let's say chromosome 21. At a SNP locus or position, each chromosome has one of 2 nucleotide possibilities, also known as an allele. Let's label these generically as allele A and allele B. Just keep in mind that A doesn't refer to the adenine nucleotide base. It's simply a generic filler to represent an allele. And at each SNP position, we can have two A's. We can have one A or one B, or we can have two B's. And across the chromosome, we analyze many thousands of SNPs, where at each position, we can determine an allelic state, whether it be two A's, two B's, or one A and one B. Now, looking at this entire chromosome as a unit, for the purposes of illustration, we can turn it onto its side. And in a normal genome, at each position, we will have 100% allele A, 50% allele A, or 0% A. Now, plotting this out as a ratio of A with respect to the position on the chromosome, we can have for these three SNPs that are displayed here, just 3 dots at 100%, 50%, and at 0%. So looking at the center SNP of this chromosome, it's 50% A. And let's say this genome now has a small contribution from another individual, the fetus. And if the fetus at this position also has 50% A, that is, it inherited one A and one B, the plotted point looks exactly the same. If the fetus inherited an A from the mother and another A from the father, then the ratio of A is now slightly higher due to the small fetal contribution of additional A. If the fetus inherited a B from the mother and a B from the father, then now the ratio of A is slightly lower due to the small fetal contribution of B. In other words, there's a little less A in that case. So now we can do this for each position on the chromosome and end up with a banding pattern due to the fetal signal. And we can now distinctly calculate what is commonly known as a fetal fraction within the context of maternal DNA. So using the information from the totality of SNPs that we're assessing, we can generate what we call an allele frequency plot. 0% fetal fraction, or no pregnancy, will have 3 bands, one at 100% A, one at 50% A, and one at 0% A. As we introduce a fetus, we can see a distinct banding pattern whose distance depends on the fetal fraction. So as you can see, the band distances increase as the fetal fraction increases. In this example, at 12% fetal fraction, the fetal bands start to appear, with the bands segregating further out at higher fetal fractions. And in this graph, we show a 26% fetal fraction. The maternal and fetal genotype combinations for each band is known and shown on the brackets to the right of the plot. Now, to illustrate what a trisomy would look like, while of course we use algorithms to make these assessments, we can also visualize it easily on a high fetal fraction sample, as shown here. So to give you a visual example, the normal 3-band allele ratio pattern, it manifests as a discrete banding pattern due to the additional contribution to the allele ratio by the 3rd chromosome. So on the left here is an example of a disomy pattern with a different pattern that's shown on the right for a trisomy. So using this SNP-based technology to distinguish maternal and fetal genotypes allows us to study cfDNA isolated from maternal blood that contains both the maternal and fetal cfDNA. We can then generate accurate fetal fraction and aneuploidy calls by analyzing the SNP ratio patterns via the advanced bioinformatics within our software. And this methodology also manifests in clinical advantages, resulting in lower false positive and negative rates relative to other technologies. Our clinical accuracy is displayed in these graphs as aggregate data from various validation studies that span over 30,000 patient samples. It's important to note that the data shown here is for all the aneuploidies of interest, and a lot of the data you'll see from various counting method companies will only show data for certain conditions like trisomy 21. However, showing data for only certain trisomies is misleading because the counting method traditionally has had issues with aneuploidies other than 21. It's also important to note that the methodology for these studies was not uniform. More specifically, some studies obtained confirmation of truth in all cases, while others only solicited outcomes in high-risk calls. Another important point is that a higher call rate is not necessarily better. One might conclude that a higher call rate means higher call success rate, but that's not necessarily the case. Just as a boy who cries wolf all the time is going to lose trust if some of his calls are false positives, a test that constantly provides calls without regard to whether the calls are confident or not will lose credibility. Our bioinformatics algorithm only makes a call when the results are of high confidence. For example, we don't call a result if the data is low confidence or has a fetal fraction that's too low. Some may interpret this as a higher test failure rate, but the reality is that more discriminate test calls means lower false positives and a greater trust in the calls. And this is clearly demonstrated by our clinical superiority that was shown in the previous slide. Since PPV is a fraction of positive calls made that are actually positive for the aneuploidy of interest, it's a good indicator of calling out the right answer. Compared to the traditional first trimester screening, where 97% of women will get a false positive and be referred unnecessarily for an invasive testing, our technology will only result in around 9% for trisomy 21. Now, just a high-level view into what our workflow looks like. Collected blood undergoes standard centrifugation technique for plasma isolation, from which cfDNA is extracted. What we call library preparation here is the preparation of fragmented cfDNA to undergo highly multiplex targeted SNP amplification of the over 13,000 different targets in a single reaction. We then barcode these samples to be able to pool and sequence them, where the barcoding allows us to demultiplex the sample after sequencing. Data analysis is performed in the cloud through our Constellation software, which generates information needed for reporting. Those who are implementing the workflow will have aid from both the technical lab aspect from our field applications team and the software aspect from our software applications team. So what does Natera offer to labs interested in genetic testing for NIPT? Natera has spent millions of dollars in research, development, clinical validation, as well as regulatory registration for its advanced technology, and is committed to continually improving and innovating genetic testing technologies. Natera has made the decision to make these technologies available to lab customers who may be interested in using these technologies to kickstart or extend their genetic testing capabilities. And Natera's technology offering includes the bioinformatics software platform, the proprietary reagents, and support from our applications and our support team, which allows labs to easily develop, validate, and clinically offer genomics assays, namely the Panorama assay at present, with future applications in liquid biopsy for cancer screening using the same Constellation software platform. An incorporation of Constellation will allow the lab to grow their testing options alongside Natera's increasing portfolio. We have readily available reagents for library preparation that streamlines cfDNA preparation workflow, as well as for the multiplex PCR for simple targeted amplification of the over 13,000 SNP targets within a single well per sample. The Constellation Bioinformatics platform accelerates genetic testing by combining innovative science with scalable bioinformatics. For genomics labs that are looking to enter or expand genetic testing, Constellation powers highly accurate, proven science that can quickly deliver answers with confidence. In fact, this platform houses the same algorithms used by Natera's CLIA facility and offers many other benefits, including HIPAA compliance, basic internet requirements, and foundation for implementing future genetic tests. And last but not least, we have a dedicated field applications and software application support teams to aid with any questions, provide guidance, and offer troubleshooting assistance. So that concludes the presentation portion. Thank you, everyone, for joining me today. Now I'm going to turn it over to Rupert to see if we have any questions from the audience.
Thank you, Sam, for that informative presentation. For more information on this webinar and some of the content that Sam has provided, please click on the link section to review or download materials. Now, before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. So the first question, how is your NIPT technology different compared to other NIPT technology?
So other technologies use counting, where genetic material in the blood is analyzed for chromosomal abnormalities without differentiation between what's maternal and what is fetal DNA. In our technology, carefully selected SNPs are sequenced, and this allows us to differentiate between maternal and fetal DNA and assess chromosomal abnormalities. This provides us a unique capability to determine fetal fraction and improve clinical performance.
Great. The next question is, what does it mean when you say you can differentiate between mother and baby DNA? Don't all NIPT do this?
So not all technologies have this capability to differentiate the mom and baby's DNA for assessment of genetic abnormalities. In the case of counting, there's no clearly discrete way to differentiate the mother and baby's DNA. Using SNPs, though, we can tell the baby apart from the mother due to differences in the inherited SNPs. We can also determine how much difference there is, which allows us to calculate the fetal fraction.
Thank you. And the next question is your reagent kit or bioinformatics CE marked?
Yes. So we offer a cfDNA library preparation kit, a multiplex PCR kit, and the software cloud platform that are all CE marked.
Excellent. Our next question is, where in the world is your technology available for licensing?
So we'll be happy to work with you to discuss availability of licensing at your location. Please contact us after the webinar if you're interested, and we will be happy to get in touch with you to discuss further. You can also find contact information in the link section on your screen, and we definitely look forward to speaking with you about that.
Thanks, Sam. The next question is, what is the process for bringing in your technology to our lab?
So once all the agreements are in place, our scientific and software applications team will work very closely with you to help you obtain more information, training, guidance with implementation, and offer troubleshooting as needed. And that's the high-level overview, and we can discuss in more detail if you'd like to contact us.
Thanks, Sam. The next question is, how is the fetal DNA distinguished from mother DNA? Is there a target sequence that fetal DNA has that the mother DNA doesn't have?
That's a good question. The fetal DNA can be distinguished because we're targeting polymorphisms in the genome, and at these locations that we amplify using our highly multiplexed PCR, depending on what the fetus is inheriting from the mom and the dad, we can see if the genotype is different from the background mother's DNA, that this DNA is the fetus. So this is how we can differentiate using SNPs. What is fetus and what is the mom?
Thanks, Sam. There were several questions asking this. Which NGS platform is our technology on?
So at the moment, we use Illumina sequencers. We have some efforts in validating other platforms as well. For more information, I would say feel free to contact us, and we're happy to talk with you about that.
Thanks, Sam. The next question is, do you provide on-site training?
Yes. So we can definitely help provide training on-site as needed. If troubleshooting assistance is needed, we can help with that as well. So with the implementation process, we have the technical team as well as the software team who can also visit your site, and we're there with you each step of the way to help you through that process.
Excellent. Next question is, how does the system determine which tests are non-decisive? Is it based on how clear and separated the allele bands from the chromosome, from the fetal DNA contribution looks?
Sorry, I'm not sure exactly what that question means. What is the? Could you repeat that question?
Sure. How does the system determine which tests are non-decisive? I guess what he's trying to ask is, does the system differentiate based on how clear and how clearly the bands are separated?
So with the SNP plot that I had showed earlier, we can visually kind of estimate the fetal fraction. However, our software has algorithms that will calculate this for you. So there's really no visual component in stating what the fetal fraction is. We have algorithms to do that for us. However, if we were to look at the SNP plot, we could visually estimate what that is, depending on how far the band is away from the maternal bands. I hope that answers the question.
Sure. Next question is, when will your technology be available for licensing, I guess?
So the technology for NIPT is available now. Depending on your location, we would have to discuss further with you. So we have a couple of emails here listed on the screen, and you can also check the links. And feel free to contact us when we can discuss that in more detail with you.
Excellent. The next question is, how did you choose the 13,000 SNPs?
That's a very good question. So first of all, we needed to choose SNPs that are located on the chromosomes of interest. So we're looking at SNPs on 13, 18, 21, and the sex chromosomes. The sex chromosomes are a little different, but for simplicity, we'll say these are all SNPs. And the SNPs that we choose, they have to cover the length of the chromosome, just so we have a good idea of a good picture of the entire chromosomes that we're interested in. We also have to choose SNPs that have high minor allele frequencies. So there's a higher chance of difference between the mom and the fetus, so that we can actually make good determinations. If the baby's DNA is the same as the mom's DNA, that is, if the genotypes of all of these SNPs are the same, then since there's no difference, we can't tell between them. However, we've carefully chosen these SNPs that have high minor allele frequencies. That is, they have a high chance that the genotypes are different between the mom and the fetus, so that we have a very good idea and a very good chance of actually determining the difference between the fetus and the mom.
Excellent. The next question is, what annual volume makes economic sense?
So this depends on many factors, and I think this is a question that could best be addressed probably by sales, and we do have a sales team that can work with you closely to help work out those numbers with you. And again, you can contact us. We can provide you with more details about that.
Great. The next question is, does your technology involve detecting carriers of thalassemia using miniseq and analysis using bioinformatics software to detect new mutations?
The NIPT does not look for this. So we have a set panel that looks at certain chromosomes, and we're looking at aneuploidies with extension of microdeletions as well. As for the mutation you're talking about, it may be covered in some of the other products. However, I would say to get more information about that, please contact us. We would have to have someone with more expertise in the other product lines to help you with that question.
Excellent. And we have time for just one more question. To distinguish which SNPs belongs to mom or fetus, wouldn't you have to know the sequence of the mom first?
Yes, and we can tell what the mom's genotype is at all of these SNPs. Within the blood, so from the plasma from which we isolate the cfDNA, that contains both the maternal portion as well as the fetal portion. And we can determine what is the maternal background, and from there we can also see what's different, and what's different would be what's coming from the fetus. So we do get a picture of what the mom's genotype is at all of these SNPs, and at the places where they're different, that's where we can tell the fetal genotype. And depending on how different they are, the proportion of the fetal DNA to the maternal DNA, we can determine fetal fraction, so the signal coming from the fetus. And based on all of these differences at all of these SNPs that we're interested in, we can determine chromosomal abnormalities as well.
Thanks, Sam. Due to time, we won't have time to go through all of the questions, and I encourage the audience to follow us on social media, Facebook, LinkedIn, and Twitter, where we can answer all those questions there, and you can follow us there. So once again, I'd like to thank Sam for his presentation. And Sam, I'd like to ask if you have any final comments.
Yeah. Thanks, Rupert. I'd like to add that while Natera has invested millions of dollars in R&D and clinical trials, and we're at a point where we believe these technologies are invaluable to all who may benefit. And as such, we're making our technologies available to anyone who is interested in bringing the most advanced genetic testing technologies and algorithms into their labs. The audience members can click on the links on your screen to get more information on how to access Natera technologies. And with that, I'd like to conclude this webinar, and thank you very much to everyone for joining us today.
Thanks, Sam. I'd also like to thank our sponsor, Natera, for making today's educational webinar possible. Today's webinar will be available for on-demand viewing through 6 weeks from today. You will receive an email from Labroots alerting you when this webinar will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. And until then, see you next time. Thank you and goodbye.